All cells have cell membranes to help control what enters and exits the cell. Otherwise, harmful things like viruses or toxic chemicals could easily enter the cell and cause damage or chaos. Likewise, cells would have trouble keeping and holding on to things that it needs, like water, proteins, and organelles. Cell membranes are what we call selectively permeable, which means they let some molecules pass through them but prevent others from passing through. But how can cells take in molecules that are too large to diffuse through the cell membrane? Similarly, how can cells get rid of large molecules that it doesn't need anymore? Cells have solved this problem through the processes of endocytosis and exocytosis. Let's start with endocytosis. When a cell recognizes a molecule or group of molecules to be taken into the cell membrane closest to those molecules begins to fold inwards. As the molecules approach the cell, the molecules enter the groove or furrow caused by the folding cell membrane. When the molecules have been almost surrounded by the membrane pinches off the final side, creating a small spherical membrane called a vesicle that completely surrounds and encases the molecules. These spherical membranes are called vesicles. The membrane of the vesicle can then enter the cell and be disassembled, releasing the molecules. The cell can go about using the molecules for whatever it intends. One example of endocytosis is the absorption of iron. Iron is carried to the cells of the body attached to a carrier protein called transferrin. When cells detect transferrin, they absorb it using endocytosis. Once inside the cell, the transferrin releases the iron, which can then be used by the cell. Another slightly different example of endocytosis is shown by white blood cells. White blood cells are part of the immune system, as they help identify and destroy foreign microbes like bacteria. When a white blood cell identifies a bacterium, it uses endocytosis to swallow the bacteria cell. Then, the white blood cell can use enzymes to destroy and dismantle the bacterium before it has the chance to cause harm. When cells use endocytosis for solid matter, like bacteria, it's referred to as phagocytosis. Endocytosis of liquids, such as oil droplets absorbed by cells in the intestines, is known as pinocytosis. Exocytosis works in just the opposite way. If a cell has waste matter to get rid of, first it packages the material into a vesicle. The vesicle travels to the cell membrane, and the vesicle membrane fuses with the cell membrane to force the material outwards. This can be used for waste, but is also often used to secrete important chemicals from one place to another, like enzymes, hormones, or antibodies. A cell's ability to perform these two processes allows cells to absorb substances they need to survive and to release waste substances that would disrupt their metabolism. They also enable cells to use chemicals called hormones to communicate with each other chemically, which, in so many ways, allows cells to work together.